or cloud computing platform that we used in the study. These are the vegetation indices that we derived from the satellite data, and this is the descriptive statistics that we developed. But most importantly, I just wanted to show you that our data was not or the distribution was not well distributed, so it was somehow positively skewed. So it somehow affected our results. And these are the correlation results that we developed in terms of estimating leaf area index as well, using bands and indices. So these are the bands that were found to be good in estimating leaf area. The one with a cross are those that were not good. The one with a right are the ones that were good. And on the far right, I don't know if it's your left, you'll see the indices. Those were also good when we actually compare them with leaf area. We have also developed some scatter plots to show you the models that performed well between stepwise multiple linear regression as well as random forest. And from all of them, we found that the random forests have performed better, of which performing or being able to actually explain at least 81% of variation when compared to other models. And in order to find the optimum or important bands for model development, we found that only band 4, band 11, as well as 12 were found to be good. You can see from that particular red line there that it cuts across those that are acceptable for model development and those which are not. And this is the map that we created based on the optimum regression model. And it shows the spatial distribution of leaf area index across the field. And what next? What next is that this can be then used and be included in a computer to do what we call precision agriculture and be able to navigate the areas that requires intervention and be able to increase productivity in the tomato fields. And the take-home message that I wanted you to get is that satellite data can be used to estimate leaf area, and cloud computing platforms are very efficient, especially by replacing the traditional methods, because of cloud computing platforms actually increases efficiency, and you can be able to work with big data, which they've been talking about since the morning. So thank you very much for listening to me. I'd like to acknowledge the following people.
Thank you very much, Mashau Frederick. We have went aboard by 1 minute, so that consumed the time for the question. Can we have the questions and comments for Mashau?
Okay. I have just a quick one. Since you use different models, what was your goodness of fit test? How did you choose the best model from the models?
The best model was actually selected based on root mean square error as well as the R squared. So those were the two methods that we used to actually validate the model to see if can this model be used to actually estimate leaf area index.
Thank you very much. Just out of curiosity, can RF also work well when you're doing evaluation of drought stress?
Actually, RF is a very good model, a machine learning model that is now most of the scientists are actually recommending, because RF can serve as a regressor as well as a classifier. So you can, instead of like using your normal regression, you can use your random forest for that in order to maybe establish relationships between variables. And the good thing is that you can be able to work with big data. It does not suffer from like issues like multicollinearity as well as saturation and stuff like that. It's actually highly recommended as one of the machine learning and artificial intelligence algorithms.
You mentioned the issue of skewed data. How do you propose to handle that?
Normally, when you're working with maybe regression, if the data is skewed, then you'll have to transform it. But because of we are working with machine learning. Machine learning seem not to actually really suffers from things such as data, the data that is not normally distributed. So that's why if you check my models, the stepwise multilinear regression model performed worse. Maybe because I should have maybe transformed the data so that it can also maybe try to compete with the random forest.
Yeah, okay.
Thank you so much, Mr. Mashao, for your presentation. Just a quick one. So you said band 11 and band 12 they were the most important bands in estimating leaf area index. What do you think is contributing to that? Like, why is band 11 and band 12? Do you think they are really relating to leaf area? Yeah,
That's a very good question, but the most important thing that actually contributed to band 11 and 12 having the best is because of the flowers. Remember, I stated that I collected that data during flowering stage. So under normal circumstances, you wouldn't expect like band 11 and 12 to behave in that particular manner. So because of flowers, so I can recommend that if, let's say you are working on crops that have flowers, those are the bands that you can use.
Okay, thank you very much, Mashao, for the question and the discussions. Further discussion, we can take them over lunch and beyond. Can we give Mashao a round of applause as we call upon Sateche Tabiso? That's also a staff member. Tabiso will talk about the effect of canning on sweetness of underutilized Transvaal red milkwood fruit. Over to you, Tabiso. So that there is no confusion, I'm only allowing them because they they're the staff members. They want to sit here, stand here. But otherwise, all our presentation are taking place from the floor for obvious reasons that were explained in the beginning. Thank you so much. Whilst they're figuring this out, can I check if Victoria Tokwa is the presentation loaded? Okay. Madimeja Mutako.
It's fine.
Yeah, let me move it and see. Yeah, no.
Okay. Happy Bongo. Bango, sorry. Isaac Lizwalo. Kulufelo Lizong. Okay. Thank you so much.
Thanks very much. Good day, ladies and gentlemen. I'm Tabiso Sateche. I'll be presenting study titled The Effect of Canning on Sweetness of Underutilized Transvaal red milkwood, scientifically known as Membersto zihiri. The study was done by myself, Prof. T.P. Mafio, and Dr. Mataba. Ladies and gentlemen, Membersto zihiri, it's an indigenous crop or a fruity crop that is found in Southern Africa. And study has been done around 2000 by Prof. Molel and Prof. Mashela, who indicated that amongst the investigated indigenous crops in rural areas, Membersto zihiri was one of the mostly liked fruit. And this might be due to the fact that it has more of biotic compounds. It has a very good vitamin C, and it's also a very delicious crop, which is normally eaten fresh. So, because of this indigenous crop, the availability and also the consumption pattern of it sometimes is not good because of one, shelf life, after harvest, crop will obviously undergo through the process of respiration, which obviously will reduce the shelf life. But because of the fruit being found in the wild, the seasonality of the crop is also a factor. Therefore, it's important that, because of that seasonality, it's important that we find methods that would preserve whatever that is available, especially because of the erratic rainfall and all those factors that might contribute to fruit not rather the tree not flowering. So as a result, a method such as canning could actually be used to preserve the fruit, maintain the freshness, and the canning process or technique is actually effective method because of the brim or the so-called syrup that is normally used. That is basically it reduces the microbial contaminants and then it maintains the freshness of the fruit. So this is basically the crop the fruit that I'm referring to, membersto zihiri. And like I said, the crop is mostly liked and is eaten fresh. So because of the reduced shelf life, as I explained, due to the respiration rate, this obviously presents a challenge in terms of making sure that the fruit is available probably throughout the year. And as a result of that, there is of course a need to make sure that we maintain the fruit for long in terms of availability, but also this will also help in terms of consumption pattern, because obviously people will not eat something that is not there. So once you increase the availability, you actually might increase the consumption of that particular crop. So in terms of the gap, a lot of work has been done on other methods, especially the pre-harvest, the propagation, which has been done by Professor Mashela in the university, but also the harvesting method, which actually was done by our group, where we've actually determined the perfect harvest stage and also those indices that could actually be used for harvesting. Now, as I've explained, due to the biotic compounds that the crop has, it is actually has ability to somehow reduce poverty and malnutrition in the country. And as a result, there is actually a need to make sure that we preserve the crop so that people can actually get it if they want it, especially among the rural communities. Therefore, the results of this are that the objective was to evaluate the effect of canning treatment on sweetness of membersto zihiri fruit. And this sweetness obviously determines the quality, because like I think you know, most of the people would want the fruit that is sweet as opposed to the one that is sour. So this is actually a determining quality factor in terms of acceptability by consumers. Now, we harvested fruit from Bochum. Obviously, like I said, the fruit is found in the wild, so we got it from Bochum. And thereafter, we sorted them based on the uniformity of the maturity. Obviously, it will be the color. And we've also made sure that within our experimental unit, we do not have crops or fruit that has diseases or any damage, but also we classed them according to the uniformity of size. Now, the total soluble solids of our experimental unit and also the pH was, the total soluble solids was around 2% and the pH was around 5. And this was done before processing. The experiment comprised of 2 treatments, which we had non-canned. The non-canned were actually stored at minus 72 degrees Celsius, and then we also had canned. So the canned, we used the syrup, which was we actually used the sugars and the lemon juice, and the pH of the syrup was about 4%, which is actually what is recommended in the processing industry. The pH would obviously facilitate in terms of reducing the microbial contaminants. So we made sure that our syrup had 4 in terms of the pH. Then after that, we then subjected the experimental unit to the thermal process, where now we made sure that we boiled the fruit, which obviously had the syrup. We boiled for 30 seconds, and thereafter we stored them for 6 months at ambient temperature. And this is basically what we did. And then after that, post-canning, we actually determined the total soluble solids, which is normally used to indicate the sweetness of the fruit. We also determined the tratable acidity, which in this case we transformed it into the citric acid equivalent. We also determined the ratio of the TSS and the TA. We then determined the prim A, which is actually sweetness minus acidity, which is usually used, it actually determines the sensitivity of the tongue to the total soluble solids compared to the sweetness. And then because we had two treatments, we then used the student t-test, and the GenStat software was used for us to be able to determine whether indeed canning had effect. So basically what we found was that the total soluble solids increased in the canned fruit compared to the nut, and the same was also found in the TA, the total tratable acidity. And the TSS, the reason it might have increased it because of probably the syrup, but also, as you know, during post-harvest storage, you would obviously have a transformation or the starch hydrolysis where you have an increase of sweetness. So the TSS might have actually be the equilibrium between the sweetness from the sugar from the fruit and also the syrup. The TA might have also increased due to the fact that we made sure that we used, in our syrup, we used the lemon juice. As we know, lemon is a citrus group, therefore it obviously has a high amount of citric acid as organic acid, so it might have also contributed to that. But the TSS, looking at the TSS, it might actually indicate that the fruit was sweet. But the TSS, the problem with it is that even though this variable indicates about 80% of the sweetness, the problem with it is that it contains organic acids, phenolics, and other compounds that might actually contribute, especially the refractor. Those antioxidants, they can actually reflect the light of these compounds that I've just mentioned. Therefore, even though the TSS have increased, that might actually not be true in terms of the sweetness of the fruit. Now, because of that, we measured the TSS and the TA, which obviously would have a problem, even though you can see that the non-canned had about 20%. That might also be a problem because of the denominator and the numerator actually has both acidity and it has the acidity and the sweetness. So the TSS might also not, the ratio might also not be a good indicator of sweetness. So what is found to be a very good indicator of sweetness would be the brim, which obviously is sweetness minus acidity, and this variable, as we see, there was no significant difference. This means that the canning process actually has no negative effect, no positive effect on the fruit, which is good because we don't want anything that will actually transform the true state of the fruit. So in conclusion, we found that this canning method can actually be used to preserve the fruit, membersto, but as you have seen with the results, we did not do the sensory evaluation, which is something that we actually do in order to get the perception of the sweetness from this technique. Thanks very much.
Thank you very much, Thabiso Sateche. Can we have comments and questions for Thabiso?
Thanks very much, Thabiso, for insightful presentation. Thanks for the good work that you are doing, colleagues. Mine is just go back to the issue of, the nutritive value of whatever that we are doing. If you look for, I think there was a guy who have presented also with the same red milkwood. So this seems to be one of the most crop that we have to take care of. And especially that most are the wild food crop that are available in the country. So I just want to check with you, you indicate that there is a potential for this crop to be, cultivated. So in your, I just want to get surety here, whether people are, is there people who are actually planting this at their home, because I can see that we have collected some of Sasambul and Bochum, I know where they are available mostly in the mountains. But at home, is there people who are actually have this milkweed?
Thanks very much for the question. Yes, especially in Bochum, where we sampled, there are a lot of households where they've actually planted it. So they do plant it. Also, actually, it has a potential in terms of propagation. Like I said, work done by Prof. Mashela in our university has actually demonstrated, or they've actually came up with a protocol for the propagation of the crop. So it actually has a potential to be taken further into commercial production. Thanks very much.
Okay, so just into checking the bottling part and the boiling. Maybe I missed you because I was. Were you boiling into them already canned, or were you boiling them first and then can them after? I just want to check, because if, say for instance, if you wanna maybe check whether you'll maintain the sweetness of that particular, but it will be poisonous. No, I'm dreaming now. It will be poisonous. Yeah, no, just to clarify that, because.
Okay, so what happened is that we disinfected the fruit. Obviously, that would be through the sodium hypochlorite. We applied that. After that, like I said, a thermal process. So basically what we want to do there, because we have a syrup that might actually be contaminated by microorganisms. So if we apply a thermal process, that will obviously, through the syrup, that would facilitate the killing of those microbials. So you'll not have microbial contamination from the bottle.
Okay, thank you, Tabisa, for the presentation. On your rationale, you say that the fruit could be used to reduce poverty and malnutrition. How?
Like, thanks very much for the question. I'm not into economics, but my understanding is that if you people are poor, they normally eat. So, I mean, if they are malnutrition, malnutrition or whatever, something like that, so they would eat. Obviously, if I'm saying I'm poor, it means I don't have access to adequate food or the food that is healthy. So the propagation of the crop would actually increase in terms of the food, rather the food that is available for people to be able to consume. So that would obviously contribute in terms of poverty reduction. But like I said, I'm not into economics, so I might not really delve much into.
Okay, so what are the nutritional values of this fruit?
Like I said, it has a vitamin A, rather vitamin C, which is obviously a very good compound for the healthy or for the body, but also there are other bioactive compounds, like for example, your carotene, your beta-carotene, they also contribute in terms of health. So, but of course, because it's an indigenous crop that is understudied, hence the title underutilized. So there's still a lot of work that needs to be done in order to determine the bioactive compounds and also the bioaccessibility of those compounds, because most of them, they are bound by cell membrane, so they might not be available, even if you find that they are available in the crop, but they might not be available for the body.
Okay, thanks.
Thank you so much. Can we give Thabiso Sateghe a round of applause? We have exhausted all the time that was allocated for this presentation. We're moving right along. Victoria Tlhoke. This is a master's research on the prediction of egg weight and ovarian follicles morphology of Potchefstroom chicken breed. Thank you. Over to you, Victoria.
Good day. My name is Victoria Hlokwe. I'm under the supervision of Prof. Diasi and co-supervision of Prof. Mbazima and Dr. Gunya. I'll be making a presentation under the topic Prediction of Egg Weight and Ovarian Follicles Morphology of Potchefstroom Kokok Chicken Breed. Okay, Potchefstroom Kokok is just an indigenous breed that is used by the farmers for the production of both meat and eggs. And the breed is characterized by its black and white feathers and yellow feet, as displayed in figure 1. Can I start?
Yes, please start. Go ahead.
Okay, I'll be making a presentation under the topic Prediction of Egg Weight and Ovarian Follicles Morphology of Potchefstroom Kokok Chicken Breed. Next slide. Oh, sorry. Okay, the slides aren't moving. I don't know, they're not moving.
Can you?
Do it.
Do it.
Do it, let's
Test it.
see. We've put it back. Yeah.
Okay, sorry about the glitches, Victoria. We apologize for that. We're no longer going to allow anyone to present without using that because it will create trouble for us. Thank you so much. Go ahead. You can start. You have your full 10 minutes.
Oh, okay. Okay, good day. My name is Victoria Hlokwe. I'm under the supervision of Prof. Diasi and co-supervision of Prof. Mbazima and Dr. Gunya. I'll be making a presentation under the topic Prediction of Egg Weight and Ovarian Follicles Morphology of Potchefstroom Kokok Chicken Breed. The slide there displays the presentation outline. Potchefstroom Kokok is just an indigenous breed that is used by the farmers for the production of both meat and eggs. And this breed is characterized by its black and white feathers and yellow feet as displayed in figure 1. Sorry. And then the preovulatory ovarian follicle is just a sac that is containing an oocyte just before ovulation. And this sac is containing some necessary fluids that are important for the maturation and the maturation of the follicle just before ovulation. Due to an increasing human population, the demand for eggs becomes very high. And when the demand is high, there is a need for improved production in order to meet the demand by the population and to remain food secured. However, this Potchefstroom Kokok chicken is having low egg production in terms of the egg quality traits, the egg weight, and the egg number, and that is affecting the profit margins of the farmers who are farming with it. Hence, the egg quality traits and the egg weight of this breed require improvement. Studies conducted previously, they found that there is a link between the egg weight and egg quality traits, and those studies were conducted in 2018, 2020, and 2016 in the mentioned breeds. The studies conducted in 2017 and 2022, they found that there is significant difference in the average number and weight of the small and large yellow follicles, and those papers were done in the breeds mentioned there. However, the ovarian follicles morphology of Potchefstroom Kokok chicken breed is not yet known. The aim of the study was to evaluate the association between egg weight and egg quality traits and ovarian follicles morphology of Potchefstroom Kokok chicken breed. And the objectives of the study were to determine the association between egg weight and egg quality traits, and lastly to identify the ovarian follicles morphology of Potchefstroom Kokok chicken breed. This is the hypothesis of the study, which is in line with objective 1. The study was conducted at the University of Limpopo Experimental Farm, and other parts of the study were conducted at the Animal Laboratory at the Department of Agricultural Economics and Animal Production. A total of 50 Potchefstroom Kokok chickens were used in the study to lay eggs for the conduction of the study, and these hens were raised under intensive production system according to the normal husbandry procedures of the paper published in 2012, and cross-sectional study design was used in the study. 300 eggs were collected from the chickens at the age of 20 weeks for a period of 10 weeks to measure the physical egg quality traits. And in the study, the egg weight and internal-external egg quality traits were measured as listed there. And then the egg weight and the egg yolk weight were measured using the weighing scale as indicated in figure 2, and the egg length and egg width were measured using the vernier caliper as indicated in figure 3. To identify the morphology, 6 chickens were selected and they were taken to the slaughterhouse at the age of 30 weeks. They were slaughtered and then to harvest the ovarian follicles to determine the morphology. And then to determine the morphology, the sizes of the large yellow follicles were taken, and the average number of both small and large yellow follicles were taken. And then figure 4 there is showing the chicken ovarian follicles of Potchefstroom Kokok from F6 to F1, with F1 as the largest follicle, and the black arrows, they indicate the small yellow follicles. The statistical package for social sciences version 26 was used for data analysis, when Pearson's correlation was used to find the association to achieve objective 1, and the descriptive statistics such as means and standard error of means, as well as one-way ANOVA, were used to achieve objective 2. All the statistical analysis were done at 5% significance level for statistically significant and 1% for highly statistically significant. For mean separation, Duncan multiple range test was used. Table 1 there shows the results for correlation between egg weight and egg quality traits of Potchefstroom Kokok chicken breed. And in the results, 1 star means that the correlation was significant at p-value less than 0.05, and 2 stars means that the correlation was significant at p-value less than 0.01. The findings highlighted in red there are the findings of the traits which had a positively high statistically significant association with egg weight, and those traits included the egg width, the yolk width, the shell surface area, the albumin weight, the albumin ratio, and the egg volume. And such traits, scientifically, they are assumed to be controlled by the same genes, meaning that when you're improving those traits, you're also improving the egg weight as they're positively correlated. And the traits which are highlighted in blue there, they are showing the findings of the traits which had a negatively high statistically significant association with egg weight. And such traits, when you're improving them, you are decreasing the egg weight, and that is not wanted. Table 2 is showing the findings of the average number of ovarian follicles morphology of Potchefstroom Kokok chicken breed, and the findings displayed that there is a significant difference in the average number of the large and small yellow follicle, as the p-value indicates there is less than 0.05. The findings also showed that the small yellow follicles, as highlighted in red there, they had a higher average number when compared to the large yellow follicles. The table 3 there is showing the average weight of the large yellow follicles in Potchefstroom Kokok chicken breed. And the findings showed that there is a significant difference in the average weight, of the large yellow follicles, as the p-value indicates there is less than 0.05. And these findings further showed that follicle 1 there, as highlighted in red, is having a high average weight when compared to other follicles. However, it was not statistically significantly different from follicle 2. And follicle 6 had a lower average weight when compared to other follicles. However, it was not statistically significantly different from follicle 5. The correlation findings of this study, they are in line with the paper that was published in 2018 in French broiler guinea fowl, and then again they were in line in paper published in 2020 in Saguinea chickens. And the morphology findings were similar to those of the paper published in 2017 in the 2 breeds mentioned there at 36 weeks of age, and they differed from that of the paper published in 2022 in yellow-bellied chickens aged 50 weeks. And the variations in the morphology findings could be due to breed differences or the age at which the morphology was identified. And then in conclusion, the study found that there is a link between the egg weight and the egg quality traits mentioned there. And again, the morphology findings have indicated that there is a significant difference in the average number of small and large yellow follicles, as well as the average weight of the large yellow follicles. And this shows that egg yield is more dependent on the continual growth and development of chicken ovarian follicles. This study recommends that importance of egg weight be emphasized to the farmers as it affects their pricings and profit margins, and again the importance of the association between egg quality traits and egg weight, as the egg quality traits play a role in egg weight. And then further studies need to be conducted on the chicken ovarian follicles morphology in other South African indigenous breeds. I would love to acknowledge the University of Limpopo, Department of Agriculture, Economics and Animal Production for giving me an opportunity to conduct the study. My supervisor, Prof. Tiasi, for the skills training he provided through the conduction of the study. And I would like to thank my co-supervisors, Prof. Mbazima and Dr. Gunya, for the contribution towards the study, as well as Tiasi research team. These are some of the references which were used in the presentation. So, ladies and gentlemen, this marks the end of my presentation. Thank you.
Thank you very much, Victoria. May we have questions and comments for Victoria Tloke?
Okay, thank you very much. The way, you said the weight and the quality. What were you feeding them? Were they free ranges, or were you giving them what broilers are eating to get into the weight and the quality? I just want to check, because after maybe the study, you must be able to talk to poor farmers who cannot maybe buy feeds from Mido in Midrand, is it in Joburg or maybe Potchefstroom, to feed into those things. What were you feeding them? Were you feeding them calcium, whatever food, or maybe there were free ranges, or what was happening actually to get into the quality?
Okay, I was feeding them the laying mesh to get the eggs, yeah.
With calcium, the one
yes, the balanced, yeah, it's a balanced diet, yes.
Thank you very much for your very enthusiastic talk. And afterwards, I would like you to come and I'll teach you to say koek koek.
Myself, I have a very quick one. On the post-hoc test, why did you choose Duncan's?
Why did I choose?
Duncan's range test.
Oh, okay. There's no specific reason why I chose Duncan multiple range test. I just chose it as one of the immune suppression tests, the post-hoc.
Okay. Okay, thank you for the nice presentation. I just wanted to know the type of eggs you used. Were they fertile eggs or just the eggs, the table eggs?
Oh, they were not fertile. It was just the table eggs, yes.
Okay.
What 50 chickens you took.
70? Why not 30? Why not 40?
Oh, okay, yeah.
You have to give a reason that reason I think. Just tell us why.
Oh, according to when I was checking the literature, so 50 is also acceptable for this kind of study. So it was supported by literature. So I just decided to use this number because it's giving me a precise result. Yeah. Oh, okay.
Okay, thank you for the good presentation. My question is from your problem statement. You say that, okay, I can't say the name of the chicken, has low egg production. What do you think is the cause?
Oh, okay. Okay, I think maybe the cause for the low egg production, maybe it might be on the, because of the genes or something. Yeah, let me say because of the genes, because this, I think because of the genes, the genetic makeup.
Okay,
Yes.
are you doing PhD or master's?
Masters.
So wouldn't it be nice for you to find out the genes that are responsible?
Okay, thank you very much.
There's one last question from here in front. I'm going to allow that, sorry. Because of the limited time, we're not taking follow-up questions.
Just a quick one.
Yeah.
I saw you were making some comparisons between the follicular stages, like F1 going through F6. You got me lost on that one. Are these things not developing, moving up?
Yes, they're developing means we like moving up. What is the question?
Why were you comparing like the different follicular stages?
I was comparing the different follicular stages.
Yeah, I didn't follow what you were doing there.
Oh, okay. I was comparing the average number of the, in my study, I was comparing the average number of the small and the large yellow follicles, as well as the average weight of the large yellow follicles themselves. So that's what was happening in my study. I wanted to know the average number that the chicken is producing, the average number of the small yellow follicles that the chicken is producing, and the number of large yellow follicles the chicken is producing.
I will save you on time on that one. I'm still lost.
Okay, you can ask me after. I'll explain. Right. Thank you.
Okay, thank you very much, Victoria Tloke, for the presentation, and thank you for the discussion. We are moving right along. We're going to have a presentation by Madumeja Matapo. It's a doctoral study on prediction of body weight from morphological traits of South African nondescript indigenous goats of Limpopo Ngumbi municipality using different data mining algorithm. Over to you, Matapo.
Greetings, ladies and gentlemen. I am Madumeja Matapo. I will be presenting the project entitled The Prediction of Body Weight from Linear Body Measurements from Morphological Traits of South African Nondescript Indigenous Goats of Lepelle-Nkumpi Local Municipality using the different data mining algorithm under the supervision of Prof. T.L. Chiase and Dr. T.J. Mkwaba. Then, South African nondescript indigenous goats are the goats that mostly found to be kept by the rural farmers. These goats are very functional. They can survive from what the environment is providing. They are known to be hardiness and disease resistant. They play a very crucial role to the livelihood of smallholder farmers in terms of income, meat, milk, and traditional and religious purposes. So a data mining algorithm is the use of computer-based method to discover information. This data mining algorithm is used by researchers in science, health, and bioinformatics to find the association and patterns from raw data. So in animal breeding, we are using this data mining algorithm to identify traits that can accurately estimate the body weight of an animal. Live body weight of an animal is very much crucial in the livestock industry, since it can help farmers to select the best animal for during breeding selection, to determine the good dosage to administer in the animal, to provide proper feed to the animal, and also to set a better price for the animal. However, it was found that these smallholder farmers in rural areas find it difficult to estimate the body weight of their animals due to lack of weighing scales, since they don't have enough money to purchase them. So studies indicated that the morphological traits, they can be used as an alternative way to estimate the body weight of an animal. In rationale, several studies showed that data mining algorithms have been used to identify the morphological traits that can accurately estimate body weight from small ruminants. However, there is a scarce information on the use of data mining algorithm to predict body weight of South African non-descript indigenous goats found in Lepelengumbi local municipality. The aim of the study was to establish the association between body weight and morphological traits of South African non-descript indigenous goats. Then the objectives of the study were to determine the association between body weight and morphological traits of South African non-descript indigenous goats, to establish a model that can be used to estimate the body weight from morphological traits. The hypotheses were in line with the objectives of the study. The study was conducted in 3 villages found in the Lepelengumbi local municipality, which is Morotse, Linting, and Makuru. Then the animals that were used were the nondescript indigenous goats, which were managed extensively, where they were released in the morning for grazing, then recalled back later in the afternoon. The study used a cross-sectional study design. For sampling, the study used a multi-stage sampling procedure, where in stage 1, Lepelengumbi municipality was purposefully selected, since the Department of Agriculture in Limpopo indicated that it is the municipality that has got a higher number of indigenous goats. Then the second stage, we found 3 villages within the municipalities. These villages was also purposefully selected, since the farmers were found in these villages were working in hand with the extension officer in the Lepelengumbi municipalities. Then a minimum of 30 smallholder indigenous goats farmers with at least a minimum of 7 indigenous goats were randomly selected from those selected villages. Then stage 4, a minimum of 7 indigenous goats from the selected smallholder indigenous goats farmers were also randomly selected. Then the study used 700 indigenous goats, guided by the author mentioned, published a paper in 2016 that indicated that 613 indigenous goats were suitable for this kind of study. Then these are just morphological traits that were measured during the study. Then we used a Taylor measuring tape calibrated in centimeters. Then the body weight were taken using a weighing scale, calibrated in kilograms. All the measurements were taken according to the suggested author. Under statistical analysis, PSS of Vision 26 of 2019 were used. Then the Pearson correlation was used to achieve objective 1. The cut and the chart model was used to achieve objective 2. Then the goodness of fit was used to predict the performance of the models. This is a phenotypic correlation table showing the correlation between the body weight and the morphological traits of the animals. So below diagonal, we are going to find the correlation coefficient of the does. Then above diagonal is the correlation coefficient of the bucks. So the result indicated that there was firstly, the double star that we found here, they means that there is a correlation coefficient which is highly statistical correlation. Then the single star shows the just statistical significant correlation. Then it was found that the bucks we found the body length was highly correlated to body weight. Then moving to the dose, the body length was also highly statistically correlated to body weight, followed by hard girth, which was found to be just statistically correlated to body weight. This result means that when we improve the body length in bucks and does, together with the hard girth, we will be improving the body length, the body weight of the animals. Then the figure 2 shows the cat model. This model was created to come up with the traits that will be used to estimate the body weight. So this model considers the traits that effectively affect the body weight together with the other considered factors that can be used to estimate the body weight of an animal. So we were found that the body length was the one that most effectively affect the body weight, followed by the body length again. Then age was also playing a role on the body weight of an animal. Age was also playing a role, then the body length, then hard girth, and also the villages that we found the animals in. This is also a cat model. It also shows the body length as the traits that was effectively affecting the body weight. These two models were nearly identical since they showed the morphological traits that have effect and other considered factors on body weight. Then to tell them if the models performed better, we used the goodness of fit, where we were looking at the coefficient of determination. So for the model to be good, the coefficient of determination has to be high, then the coefficient of variation, root mean square, should be low. So in this case, our results indicated that the kite model was a better model compared to the cat one. The findings of the current study were in line with the findings of the author, the paper published in 2015, 2020, and 2021 on correlation. Then it was further found that the study published